Now a panel on emerging infectious diseases and the next pandemic. It's moderated by investigative journalist Sonia Shaw, author of Pandemic, Tracking Contagions from Cholera to Ebola and Beyond. The New York Academy of Medicine hosted this 90-minute event.
Okay. Thank you all for coming. I usually don't like to stand at podiums because I'm kind of short, but I'll get up on my tippy toes for you. So when I first started writing this book about 6 years ago, I certainly didn't predict that we'd actually be living through a pandemic of a new kind of pathogen kind of washing over the Americas right when the book came out. But of course, here we are with the Zika virus washing over the Americas. And ooh, that's not my talk. Hang on a second. So, over the past 50 years, we've had over 300 infectious pathogens either newly emerge or reemerge into new places where they haven't been seen before. So Zika is really just the latest one of a plethora of pathogens that have been doing this. We've had Ebola in West Africa, where it hadn't been seen before. Middle East respiratory syndrome, a new virus has come out of the Middle East. New kinds of avian influenzas coming out of Asia. Of course, a whole range of new mosquito-borne pathogens, dengue, chikungunya, now Zika, West Nile virus, etc. So the question I wanted to ask as a science journalist was, how do microbes turn into pandemic-causing pathogens? If you think about a microbe, it's just a tiny, invisible thing with no independent locomotion, and yet it turns out to in some instances, can cause this huge amounts of death and destruction. So the way I wanted to look at that was through a two-pronged approach. So first I looked at the history of one of our most successful pandemic-causing pathogens, and that's cholera. Cholera has actually caused 7 global pandemics since it first emerged in the 19th century. And the latest one is still going on right now, just a few hundred miles off the coast of Florida in Haiti. And then I coupled that with reporting from places where new pathogens were coming up, and this was work that was supported in part by the Pulitzer Center. I went to places like South China, New Delhi, and Port-au-Prince, and elsewhere to try to see how the story of cholera could shed light on where these new pathogens might be going. And what I learned is that cholera emerged in many ways in the same way that a lot of the new pathogens are coming out today. It came out of the natural environment. So cholera is caused by a bacteria that normally lives in marine habitats. It actually lives in conjunction with zooplankton called copepods, and it actually performs a useful ecological function in that habitat by helping to recycle nutrients. So places like the Sundarbans are full of cholera bacteria. The water's just full of it. This is water that's half fresh, it's half salty, and this is where all the major rivers of South Asia are draining into the Bay of Bengal. Now, for a long time, people didn't usually live in places like this, right? The Sundarbans are covered in mangrove swamps. They're tidally flooded twice a day. There's cyclones, there's tigers. So people didn't really live in cholera-rich environments. That, of course, all changed in the 19th century. The British Raj decided to turn the Sundarbans into rice fields. And over the course of the 19th century, 90% of the Sundarbans was settled. So suddenly, humans invaded cholera habitat, and this allowed cholera to spill over into our bodies and adapt to us. And of course, in our bodies, it doesn't perform a very useful ecological function, but it can kill people within a matter of hours. About 50% of people infected with cholera bacteria will die if not treated promptly. So the first pandemic of cholera started right in the Sundarbans in 1817, started to move up into Russia and out into the industrializing cities of Europe. And this is the same thing that's happening today. About 60% of our new pathogens are coming out of the bodies of animals. And it happens when humans invade wildlife habitat or we destroy wildlife habitat forcing wild animals to come into our territories. So from bats, we've gotten Ebola. From monkeys, we've got HIV and malaria, and most likely Zika as well. From birds, we've got West Nile virus, avian influenza, other influenzas, etc. And then, of course, we provide these new pathogens with great opportunities to amplify, which allows them to even better kind of adapt to preying upon human populations. And we first started doing this in the 19th century when we started living in cities, and that was a new thing to do back then. People were abandoning the farms, coming into these new cities for factory jobs. And of course, in places like New York and London, there wasn't, suburban transit systems to take people outside of the city to sprawl. Everyone had to live right in the city, near work or near the possibility of work. So in places like 19th century Manhattan, there's about 77,000 people crowded into every square kilometer in the 19th century. About 1/12 of the city was covered with cesspools and privy vaults and outhouses. And none of this was plumbed. There was no sewage system. So the typical 19th century New Yorker, because of this intense crowding, was ingesting about 2 teaspoons of fecal matter every day. And this, of course, provided a great opportunity for cholera bacteria as soon as it entered into the city to just explode. And this process of urban expansion that started in the 19th century is really reaching its peak now. So by the year 2030, the majority of the human population is going to live in cities. And they're not going to be cities like Stockholm and Washington, D.C. They're going to be cities more like Monrovia and Freetown, where there's a lot of ad hoc development, a lot of slums. About 2 billion people are expected to live in slums by 2030. And new pathogens have already started to take advantage of this process of urbanization. If you consider Ebola, for example. We've had Ebola outbreaks since the 1970s at least, but Ebola had never infected a place with more than a few 100,000 inhabitants until 2013. And within a few weeks of emerging in Guinea in 2013, Ebola had reached 3 capital cities with a combined population of nearly 3 million, which is one major reason why that outbreak was such a huge conflagration. You could also argue Zika is similarly capitalizing on urban expansion. We've had Zika around since at least the 1940s. It was in the equatorial forests of Africa and Asia, but it only rarely infected people because it was carried by a forest mosquito that mostly bites humans and doesn't bite people very much. But what we see now in the Americas is Zika is being carried by Aedes aegypti mosquitoes. And this is a mosquito that has dramatically expanded its range with human urban expansion. It loves living near people. It will breed in a drop of water in a bottle cap. So all of our plastic garbage provides perfect breeding sites for this mosquito. And unlike the forest mosquitoes that carried Zika in the past, Aedes aegypti only bites humans. But in addition to crowding our people together, we're also crowding our animals together. We have more animals under domestication right now than in the last 10,000 years of domestication until 1960 combined. Huge numbers of livestock that we're keeping right now, and many of them live in these factory farms. We have 1 million or more individuals crowded together, so they're basically the animal equivalent of urban slums. And this similarly allows pathogens to amplify and change in ways that can make them more virulent. One example of that is avian influenza, where avian influenza viruses, they normally live in wild waterfowl. They don't really make those birds very sick. But when those viruses drop into these factory farms, where all these captive animals are crowded together, they start to change, and they replicate and mutate, because that's what viruses do, and they become more virulent. And so we've had an increasing problem with these more virulent forms of avian influenza, and some of them have even evolved in ways that can infect humans. Now, we first started carrying these pathogens around in a rather efficient way in the 19th century, and that's something cholera took advantage of, and that was through steam travel. We started steaming across the Atlantic, also with clipper ships, steaming up and down all of our rivers, our waterways, and then we used steam engines to build canals. And this connected all of our waterways together, so we had this nice kind of international network of waterways. It was perfect for a waterborne pathogen like cholera to take advantage of, which it did again and again. The Erie Canal, in particular, opened in 1825, right in time to lead cholera into New York City again and again over the course of the 19th century. But of course, we do it even better today with our flight network. We have not just, a couple airports in capital cities, but hundreds of airports, tens of thousands of connections between them all. So when a pathogen emerges in one part of the world, it very rapidly can spread across the rest of the planet. And in fact, our flight network is so influential in shaping epidemics that you can actually calculate where and when an epidemic will strike next just by measuring the number of direct flights between infected and uninfected cities. This is a map done by Dirk Brockman, and he plotted that same simulated flu pandemic that I just showed you on that geographic map. But this is a map of cities as connected by direct flights, and you can see that the flu pandemic kind of resolves into this perfect series of waves. So these are just some of the ways in which the way we live allows microbes to turn into pandemic-causing pathogens. But the other big part of the story is how we respond. Because we don't just take these things lying down. We put up political defenses and medical defenses. And it's only when those defenses fail that pandemics occur. So in 1832, doctors in New York State collected this data that we mapped. It shows a pretty clear picture to us today. Cholera is coming down the Hudson River and the Erie Canal, and it's heading straight for New York City. So the obvious response at that time would have been to consider a quarantine. Close down the traffic on those waterways and protect the city. But nobody wanted to do that because quarantine was considered too disruptive to trade. So instead they said, well, the cholera, it might look like it's coming down the rivers, but actually cholera is being carried by miasmas. This was based on a 2,000-year-old theory that was medical paradigm for many years, that diseases like cholera were spread through these smelly airs that rose up from sort of decomposing organic material. They were called miasmas. And so they said the cholera is being caused by the miasmas, and then they blamed the miasmas on the drunks, the poor, and the immigrants. And those people were actually violently scapegoated during cholera epidemics in the 19th century. There was actually a company that was making money selling cholera-contaminated water to 19th century New Yorkers. So the epicenter of the epidemics of cholera in New York in the 19th century was a slum called 5 Points, which is pictured here. And it was actually the subject of the movie The Gangs of New York, if anyone has seen that, the Martin Scorsese movie. So that slum had actually been built on what was once a pond. That pond was called the Collect Pond. It was filled up with garbage, and the slum had been built on top of that. So the ground underneath the slum was very low-lying and unstable, and of course the groundwater was easily contaminated by all the leaky privies and outhouses of the slum on top of it. But the company that the state of New York chartered to deliver drinking water to the people of New York, instead of tapping clean upstream sources of water, which they knew would taste better and would be cleaner, they sank their well right in the middle of that slum. And they delivered that water to 1/3 of the people of New York. And the reason they did that is the same reason the people in Flint, Michigan decided to change their water intake, which is they wanted to save money. And they wanted to save money because they wanted to build a bank. Does anyone know the name of the Bank of the Manhattan Company today? J.P. Morgan Chase. That's right. Biggest bank in the country. So eventually, New York did move their well from the slum of 5 Points up to Westchester County and the Croton River. And cholera ended for good then. But what's interesting is why they did it. They didn't do it to protect the public health, or they didn't do it because they threw out their theory of miasmas and decided, oh no, actually cholera is carried in contaminated water. They did it because the city's brewers demanded better tasting water for their beers. Because this crappy tasting water is putting their beers at a competitive disadvantage. So I think there's a lot of modern parallels to this story today, but we can do a lot better. And the question is, will we find the political will? And that's something I hope we can talk about more during our panel and our Q&A today. Thank you so much for listening. So we have a great panel of speakers today, and our first speaker will be Dr. Lipkin. Ian Lipkin is the director of the Center for Infection and Immunity at Columbia University. And if anyone has seen the film Contagion, I will add that the scientist in that film is modeled on him.
Thank you. I'm also the John Snow Professor, which is particularly apt, given that so much of this book concerns cholera. I was told about this meeting, and I generally accept these sorts of invitations, and I was told by my assistant that I could just skim the book. And I began with that intent, but in fact I read it cover to cover. I thought it was a beautiful book. And for those of you who haven't looked at it yet, you saw a bit of an example. You have Burr versus Hamilton. You have a whole description of sort of the London Underground and the import of plumbing. You have, it's an elegant book. So what I've decided to do today is something a little bit different, although I hope during the Q&A we can talk about some of the interesting aspects of West Nile virus and MERS and SARS and other things that may be of interest to you, and talk about what I hope will be Sonia's next book. And that is the future, really, of infectious diseases, because the emphasis in this particular book is on acute diseases, the ones that obviously are associated with severe illness, pneumonia, hemorrhagic fevers, things that kill us or at least make us quite ill. And then we have these other disorders that are typically thought of as non-communicable. Now, there are parts of the book, Sonia, where you actually talk a little bit about this and the ways in which microbes have modeled us through evolution. They have contributed to our ability, for example, to become mammals by having an impact on whether or not a mother will reject the fetus. Because in fact, what is a fetus but a tumor growing inside of us? So in fact, there are retroviral elements that have been integrated in the placenta that prevent rejection of the fetus. If you look at the genomes of our genomes, they're riddled with retroviral sequences and other sequences as well. These have important impacts, but in addition, in a more basic sense, when you actually look at microbes and self, what you're really doing is examining the ability of the body to recognize something as self or not self, because essentially we are always dealing with an onslaught of things that may represent nutrients, may represent microbes, and the way we develop, the way we grow is a function of this relationship that we've had with microbes. Now, there are a wide variety of these quote-unquote non-communicable diseases, which, as I'll show you shortly, may in fact, in some instances, have an infectious trigger. And these range from coronary artery disease and stroke to metabolic disorders like diabetes, psychiatric disorders like autism, autoimmune disorders like things like rheumatoid arthritis and such, as well as many forms of cancer. Now, the number of cancers that are attributed to infectious agents may not be fully appreciated by many of you, and particularly when you look in sub-Saharan Africa, it represents up to a third of these cancers, and most frequently in people who have cancers who are under 50 years of age. hepatitis B and C, human papillomaviruses, and Helicobacter pylori. So here are some examples. Hepatitis B, which causes hepatocellular carcinoma. Hepatitis C, also hepatocellular carcinoma. Burkitt's lymphoma, which is an infectious lymphoma, also found in Africa. And human papillomavirus, which was just described only as recently as yesterday, as being largely eradicated as a result of vaccines. HIV, Kaposi sarcoma, lymphoma, Helicobacter pylori, and so forth. So as you begin to look at all these other infectious causes of cancer, as we begin to think more broadly about infectious diseases, it may be possible for us to eradicate these as well. And we know we can already do this with hepatitis B and with human papillomaviruses, but with these others as well. These are vaccine-preventable illnesses. And here was what I was just alluding to in the Journal of Pediatrics, the description of the fact that we can literally eradicate human papillomaviruses through vaccination programs, which means we can probably eradicate cervical carcinoma and in many forms of oropharyngeal carcinomas as well. Now, most of the diseases that Sonia has alluded to, like Ebola, Marburg, can probably be prevented by using a variety of vaccines. And these are diseases which are fairly straightforward to approach. Infection is likely to be implicated in a wide range of diseases of the cardiovascular system, including stroke and coronary artery disease. Inflammation is the leading contributor to these, and we now know, for example, that something as simple as bad periodontal disease can result in an increased risk of stroke too. Now, today in the Science Times, some of you may have read this article that appeared that I actually tried to sell to Donald McNeil several months ago when we started talking about autism and neurodevelopmental disorders and such. And then when we started talking about microcephaly a couple of weeks ago, I said, this is probably just the tip of the iceberg. And as we began to discuss this, I presented him with some of the data that I'm going to show you now. So this is a disorder that we tried to model in mice called Sydenham's chorea. Sydenham's chorea was a disorder that's been associated with streptococcus and rheumatic heart disease that was first described in the 1600s by a British internist by the name of John Sydenham. And what we found, in fact, is that there were a group of children who were thought to have schizophrenia but did not have schizophrenia. What they had was an obsessive compulsive disorder that was associated with these sorts of repetitive behaviors, such as you see here. And then when these children were treated with either plasmapheresis to remove antibodies or with intravenous serum immunoglobulin, you could eliminate these disorders. We were unable to identify the trigger. We replicated it by injecting these mice with group A beta hemolytic streptococci, and then went back into the children and found that the same antigens were implicated. The other thing that we decided to approach in a similar sort of way, and this was work that was done by our group in New York and by Paul Patterson, who died a couple of years ago, who was at that point at Caltech, was to look at historical associations between influenza viruses and other pandemics and stressful environments and later occurrences of schizophrenia and autism, other neurodevelopmental disorders. And what we found was that if you looked at the association of these disorders, it made sense. That is to say, schizophrenia, you would see outbreaks of what looked like outbreaks of schizophrenia some 20 years downstream, following outbreaks of influenza and other viruses. And if you look some 4 to 7 years of age, 47 years out, with autism, you would find examples there too. We were able to model this with mice, and we demonstrated that if you examine mice that were exposed halfway through gestation to a virus or a mimic of a virus or bacterial infection, it really doesn't matter. The animals would be withdrawn and huddled in a corner of the cage, as opposed to the animals which saw some sort of a normal vehicle like saline or something like that. And if you did the same thing 2/3 of the way through gestation, instead of being withdrawn like these animals become hyperactive, running all over the cage, indicating that what was important here was not so much the infectious agents per se, but the host response to infection. The other thing that we've begun to learn a great deal more about is the microbiome, and there's an excellent exhibit presently at the just literally across the park at the Museum of Natural History. And here we tried to cover through the course of this, understand something about the GI microbiome, the vaginal microbiome, the implications of this for disease. We've begun to understand now that the gut microbiome is important in tuning the immune system. It probably is important in terms of thinking about allergy. It has a role in colon cancer, obesity, cardiovascular disease, autoimmune disorders, and even depression. And the flip side of the thought of the contagion hypothesis that Sonia is talking about, the introduction of cholera and all the other things that we've seen as we've begun to clean up the environment, is that as a reciprocal relationship, we've seen that as we no longer have those sorts of risks, we're beginning to see an increased risk of atopy, asthma, and a wide range of other disorders. So it's becoming clear that we can't only think about pathogens, we have to think about balance between microbes and humans, and it's going to become very, very complex. If we look at the microbiome over the course of the lifespan, you can look at individual different types of bacteria, and the normal progression which we see here has become subverted by this modern life where we formula feed babies, we treat ourselves with antibiotics, we have obesity, we have a wide variety of other sorts of interventions which have led to all sorts of outcomes which have a tremendous impact right now on the way we live and the way types of diseases that we begin to see. If you look at the composition of the gut microbiome in an Italian city where we have what we consider to be an ideal Western diet, a Mediterranean diet, it's still very different from the fiber-rich diet that you see in Africa. And there are consequences of this. We have begun to see, for example, the appearance of diseases associated with some of these antibiotic-resistant bacteria like Clostridium difficile, which are killing people, which is, again, is sort of the wages of modern life. And we've been unable to eradicate these now with modern antibiotics, and we're beginning to use fecal microbiome transplants as a way to address these problems. And because there has been so much success with treatment of Clostridium difficile, with using these kinds of microbiome transplants, people are beginning to use them for a wide variety of other indications as well. I'm not advocating this by any means, but simply pointing out that there are people who believe that they can reverse autoimmune disorders like Crohn's disease, ulcerative colitis, perhaps irritable bowel syndrome. There are efforts to use this now to treat multiple sclerosis, autoimmune diseases like type 1 diabetes mellitus, and as well as some forms of colon cancer. So moving forward, Sonia, as you begin to think about your next book, not to say that we've resolved all of these issues with respect to acute infectious diseases, and you're going to hear much more about how we can predict them from Peter, who will follow me shortly. It's important now to think about ways in which we can bring microbes back into balance, and we can begin to understand the role of microbes and our responses to them in the genesis of a wide range of disorders, ranging from autism to cancer.
Thank you for that, Dr. Lifkin. I'm so glad my work for the next book is he's already done an outline and everything. So want to introduce Dr. Dashak, who is the president of EcoHealth Alliance. And Dr. Dashak was one of the great sources for my book, Pandemic. And if any of you pick up the book, you'll see there's a bunch of pages about him and his fabulous work, which he will now share with us. Thank you.
Thanks very much. And, what I like about this book that other authors haven't touched on is the repeated cycles throughout history that we've gone through with pandemics that we're still in. And you made the point today. We are still doing the things that led to cholera around the world. And we sort of think about these issues of overcrowded lives and non-sterile conditions as being over there. But of course, we're absolutely connected by everywhere on the planet by less than a day's flight away. And that's exactly what microbes do. They exploit new niches that we put up and evolve and adapt and move through them and infect us. So we've created this perfect system, really, for microbial invasion right now on the planet. We're in the middle of the age of pandemics. I'd like to think we're at the end of it. And I want to really talk briefly about why I believe in a more positive and an optimistic way that in 50 years or 100 years' time, future generations will look back on this and say, those guys really did have a lot of problems with infectious diseases that we've got rid of now. And let's think about the ways we can work together to get rid of this pandemic threat. Then we can deal with the other issues that Ian Litkin's discovering as we speak. So first of all, what I do, I run an organization called EcoHealth Alliance. It's a non-profit here in New York. We're science-based. And what we do is we work on emerging diseases to try and understand what drives them. What are the underlying causes of pandemics? What's the science behind it? Can we prove that something is attached to the driver of a pandemic that leads to this spread? And then can we do something about that driver? So first of all, we need to know if these really are a big issue. I mean, the book lays it out very clearly. But what's the science behind that? What's the real evidence that emerging diseases are on the rise, that we're in an age of increasing pandemics? So a few years ago, probably about a decade now, a group of us sat around and started talking about this. I'd just finished working at CDC. I was working on wildlife diseases, including a global disease of frogs, which seems off topic, but actually is an emerging disease of frogs that spread globally, that wiped out species. So even more significant than 1918 flu or cholera, it actually caused the extinction of species. And it struck me that something's going on the planet that's got a similarity between what's happening in wildlife with the rise of new diseases in those populations, what's happening in domestic animals like livestock like poultry and others, and what's happening in our own communities. And we realize that these things are connected, of course, and this is the One Health movement. We call it eco-health. We involve the environment, the ecology of the ecosystem that drives these problems. And of course, just as we've seen from Ian's talk about the human microbiome is completely disrupted by modern life, so are our ecosystems. You know, I mean, we only need to take a trip out to the Black Forest up there or Harriman State Park and say, where are the wolves? You know, where are the passenger pigeons? Where are the chestnut trees? Completely disrupted ecosystem. So what happens when we disrupt ecosystems? We also disrupt the microbiota of the animals that live in them. So back to the science. Big question a decade ago was, are emerging diseases really on the rise? So we sat around, we said, let's take every single example of emerging disease, ones that go pandemic, ones that just cause a small cluster of cases, and just get a database. This is what you do in science. You build a database and you analyze it. So we started this project. We thought we were very clever, of course, because you always do. We'll do it in a few weeks, and then we'll crank out a paper. Well, it actually took 2 years, because it turns out it's not straightforward. The data on where and when a disease first emerged, where the first case of a new disease is, are very hard to get hold of. And eventually, after a couple of years, we got this database of something like 450 emerging disease events. New pathogens, new to science, that moved into our populations for the first time, or that were already there and began to spread for some reason. And we plotted them out. And this is a plot of that decade by decade from the 1940s to the 1990s when we published it. And as you can see, it's clearly rising. But of course, so is our effort to find them. So you have to correct for that. So then we had to build a database of every single author of every paper in Journal of Infectious Disease working on infectious diseases to see how that's rising. 14,000 data points later, logged in the GIS coordinates of every author's work. We could correct this rise and show, yes, there still is a significant increase over time, statistically significant, in the number of new diseases emerging in people. So this is a problem that's increasing. Not only that, the really big ones, the ones that go pandemic, as Sonia points out over and over again in the book, are the ones that come from animals, usually. They're zoonotic. And usually they come from wildlife, like the Ebola virus, which emerged from probably bats, like HIV, which emerged from chimpanzees. So those viruses, the ones that go pandemic, are almost exclusively from wildlife, with some intermingling with domestic animals, like in avian flu. And they are the yellow bars here on this graph. They're increasing disproportionately, dramatically, compared to the rest of them. So pandemics are on the rise, emerging diseases are on the rise. The really big ones, the HIVs, the Ebolas, the 1918's flu, are increasing over time. We can actually predict how many new emerging diseases we're going to see next year, and it's about 5. And these predictions are showing to be true. And around 3 of those will originate in animals. So, that's the bad news. What are we going to do about it is the question. So we set about to look at what are the big questions to try and deal with this problem. Well, first of all, we don't really know how many viruses there are on the planet. And really to get a handle on, can we ever deal with this problem? We need to work that out. And it took us a few years to come up with a strategy. And this is what we did. We used what's commonly used in conservation biology. If you're trying to count the tigers in the Sundarbans in Bangladesh, it's really difficult to count every tiger. They don't particularly want to come out and say hello. So what scientists do, what conservationists do, is you find a tiger, you trap it, you tag it, and you release it. Then you try and catch more. And eventually, you start recatching some of those tigers. And there's a simple equation you can use to work out the size of the whole population, including the unknown tigers, based on the number of recaptures against the number of captures. So we did exactly that with viruses. And working with Ian's group at Columbia University and with our global network, our EcoHealth Alliance around the world, we started catching animals that we knew carried zoonotic viruses, these new pandemic-type viruses. And we realized if we repeatedly caught the same species over and over again and then tried to discover as many viruses as possible, we could actually do this recapture thing for viruses. And this is what it looks like. The first graph you see is just one viral family looking at the number of samples we collected. So this is about 1,000 individual fruit bats from Bangladesh. These are giant tropical fruit bats with a wingspan about this big. The bodies are about this big. I mean, they're really cute animals. I really like them. They're like puppies with wings. But the problem is they've got big teeth and a bunch of lethal viruses. So that's unfortunate. But they're really cute, and they do a lot of good in the world. They pollinate tropical trees and especially fruiting trees. So they're very important pollinators. So we went out there and we had to catch each bat individually. We had to anesthetize them individually. We have a whole team of skilled wildlife biologists and veterinarians who do this work. And then bleed them, put the samples onto liquid nitrogen in the field, which is not easy in Bangladesh. Then with an accurate cold chain, get it all the way back to our lab here, Ian's lab in Columbia University. And then we repeatedly did the very best type of pathogen discovery, something that Ian's lab is world famous for, and started discovering new viruses. And what we found was, after a while, just after a few samples, we started seeing the same viruses. So our discovery curve went up and then saturated. And then we could use that saturation point to predict how many unknown viruses there are. We repeated this for about 12 different viral families, all of the ones that cause nasty emerging pandemics in people, and came up with a predicted number of unknown viruses in this bat as about 58. Now, if you multiply, it's very simple extrapolation. It's not rocket science, but if you multiply that by the 320,000 known, the 6,000 odd known mammal species on the planet, you come up with a figure of about 320,000 unknown viruses. Now, it sounds like a lot. I mean, we only know of about 4,000 viruses so far. So there's a lot of work to do. But it's not millions, and it's not tens of millions. And in fact, if you look at the rates of viral discovery and the increased speed and decreased cost of technology to discover these viruses, we can easily achieve this. We can discover all of the potential pandemics out there. So I think this is great. And we came up with a figure because we know how much it costs to do this work. To find 100% of these viruses is about 6.8 billion. It's a lot of money. But because it saturates, if you cut off earlier, you don't need many samples, you still get 85% of them for about 1.4 billion. Now, spread that out over a 10-year period, it's $140 million a year. It's still a lot. But how much does an outbreak of one of these new unknown viruses cost? Well, the cost of SARS has been calculated. If you look at the drop in GDP of Southeast Asian countries during the SARS outbreak against the market fluctuations that you would expect, you get a figure of something between $10 and $50 billion for one single outbreak. It's absolutely mind-boggling. This is one single person in a market in China who brought bats in, because it's a bat-origin virus, and put them in a cage, and they infected other animals and people and led to a global pandemic. The real cost was because it disrupted the trade network that we rely on for our globalized pattern of travel and trade. So these things cost so much. If we wait for them to emerge, I just think we're missing the point. So what I'm calling for now is a global virome race to the moon approach to find as many of these new viruses as we can using the very best technology we've got. And let's save ourselves some money as well as many, many thousands of lives. Now, the other big question we have is, well, where will these viruses originate? Where will the next pandemic emerge from? And again, 10 years ago, that wasn't known. Everybody kind of knew. There were lots of maps out there that people drew where they put pins on, Ebola, HIV, SARS, all the rest of it. But we didn't have science to back that up. So we set about trying to do the science. We had a big problem. We had our database of every known emerging disease. We knew where and when the people who worked on them were, so we could correct for the reporting biases in the database. So we know that there's a lot of people in Europe looking for infectious diseases. So if a disease emerges in Europe, it's going to be reported. So when you look at the raw data, you see a lot of European outbreaks, a lot of North American outbreaks, but we have to correct for that. So we did that in our analysis. The other big issue is we knew that these pandemics tend to originate in wildlife, but at that time we had no idea whether one species of wildlife carries the same number of viruses as another. So we made the assumption that overall, most animals will have roughly the same number of viruses as another species. And we had incredible data on every single mammal species on the planet, the range of that species. So we knew where every mammal species was. We made an assumption that we don't need to know how many viruses it's got. We just need to know relatively how many, because we need to know relatively where the risk is highest. So we got around that problem, and we're able to show really convincingly that there's a correlation between two things that tends to be the driver of emerging diseases. First of all, where people are on the planet. And it's not just where the populations are densest, it's where we do the most to the environment. Land use change is a correlate of emerging diseases. Changes in population density are a correlate of emerging diseases. It's disruption to the environment that drives this. And the other big issue is where wildlife are on the planet. So when you look at the hotspots for emerging diseases, which this map shows, where the next pandemic will most likely originate, you see that it includes areas where there's high biodiversity and growing human populations, expanding into those areas, hunting wildlife, farming animals next to them, bringing them wildlife into markets, trading them, and generally disrupting that ecosystem balance. So this is where we work now. We work only in hotspot countries. We focus on communities who are at risk on the edge of the forests, people who work with wildlife in markets, people who work in farms. We interview them about their habits, we take samples from them, we look for the evidence of these viruses that we find in wildlife spilling over into people. And we also sample wildlife from all these hotspot countries and work within to find what those new viruses are and what the risk of those becoming pandemic is. And recently we were able to show really clear that in parts of southern China, there are still bats present that still carry viruses almost identical to SARS. There are still people there who hunt bats, who live near bats, who eat bats, which is how SARS originated. So we're able to show the clear and present danger and now work with those populations to try and change behavior and make the things they do a bit lower risk of sparking off the next pandemic. Now, I think all of this is positive and great, and I think we will crack this pandemic problem, but it's not going to be straightforward because we have to deal, as Sonia says in the book, we have to deal with some of the underlying issues that are very hard to deal with. Global equity, poverty, food supplies, clean water, really simple, well, not simple, but not high-tech issues. And yet at the same time, we in the West tend to look for a high-tech solution, a vaccine, a drug, a new test kit, whereas perhaps what we really need is to reassess, just in the way Ian is with our own microbiome, reassess our relation with our ecosystem and try and alter the balance of that and bring it back to where it should be. Thanks very much.
Thank you so much. So next we have Carl Gerstorfer. Did I say that right, Carl?
No, but it doesn't.
I'm sorry. You'll have to say it again to get it right. Carl is just back from South Sudan, and he's going to talk about his work with Ebola and some shots from a great film he's done on HIV too, right?
Yeah, I spent like the last
Origin of
couple of
HIV,
years doing two films. My last film was about the Ebola outbreak in Liberia. I was there for 2 months, and I looked at how a community experienced that outbreak. And previously, I did a documentary on the origin of HIV, and in this documentary I tried to piece together where this virus came from and how this virus actually managed to cause a global pandemic. And in that talk I will touch a lot on what Peter has been saying about the geographic origin of a virus that caused the pandemic, HIV, and what conditions there were. So it's like a bit like looking through a magnifying glass, how it feels to be like there, and what conditions are there on the ground. HIV, as you know, is one of the worst pandemics we have experienced in recent history. And HIV is a virus that comes from chimpanzees. So the pandemic form, HIV-1M, is a virus that is a similar form is found in chimpanzees. And since the 1980s, we have been knowing that it comes from chimpanzees, and scientists try to figure out where exactly this virus originated. Where did it jump from chimpanzees into humans, and how did it subsequently spread? You have to know a little bit about chimpanzees. Chimpanzees live in rainforest areas in West and Central Africa. And the good thing about chimpanzees is they don't move a lot. They don't like water, they don't cross rivers, so over many generations they stay in the same place. So what scientists did is they basically looked for chimpanzee populations, they collected their fecal samples, they looked at whether they were infected with the chimp version of HIV, and they compared it to the human version that caused the global pandemic. And by measuring the genetic distance, they could figure out where this virus must have originated. And that search brought them to Central Africa, to Cameroon, which is here, and more specifically to an area in southeastern Cameroon, bordering 3 countries, the Central African Republic, the Republic of the Congo to the south, and here Cameroon. Now, this is a very, very remote area, and we went there for our film to document the work of the scientists there. But also there's a lot of science going on looking at what Peter has been talking about, looking at the emergence of new viruses, zoonotic transmissions, because of a lot of the factors and the drivers that drive these viruses into human populations are at work there. So it takes you from Yaoundé, it takes you like 4 days with a car to get there. It's a long journey, and then you end up at this place. It's the Sangha River. It's a riverine network that flows south to the Congo, and the Congo then flows to Kinshasa, the big capital of the Congo. Now, this area has been an area that has always been remote. But since the colonial opening up in the 1850s, there was a lot of input of people and a lot of economic activity. And we know from genetic dating estimates that HIV originated in that area around 100 years ago, between the 1880s and the 1920s. Now, if you look at the history of the place, if you look at all the geography of the place, you think, why there, why then? But then only then looking at the historical development, then everything starts to make sense. Because all the major colonial powers were operating in the area. They went in there mainly for rubber, but also for timber. So there was a huge disruption going on of the original lifestyle, a huge movement of people going on because they had an enormous demand for workers. And then, of course, over the decades, there were networks being built, riverine networks, also railway networks, and you had growing cities like Kinshasa in the 1930s. It was exploding. So you had all the drivers there in place that HIV, once it managed to jump from chimps to humans, could expand and could gather the momentum to then finally make the lead out of Africa and into the rest of the world. And we also know by now that by the 1960s in Kinshasa, there was already an epidemic going on of HIV/AIDS. There were hundreds, if not thousands, of people infected. But of course, because of the post-colonial chaos that was there in the 1960s and 70s, the health infrastructure collapsed, there was a civil war going on, nobody noticed. And then it wasn't until the 1980s that we took notice of HIV here, of course, in New York and on the West Coast. So Cameroon was the place where HIV-1M originated. And as I said, we went there because we wanted to understand whether this place still today has the potential to basically breed another virus or facilitate another zoonotic emergence of a virus. These are the scientists collecting the fecal samples from chimpanzees, which are later analyzed in labs here in the US, in Philadelphia, for the HIV genetic analysis. But also in that area, and I told you it's a very remote area, it takes like 4 days to get there, and it's a rainforest area. But there are huge logging operations going on. And it's not going on, funnily enough, at the edges of the forest, but it's going on in the middle of the forest. So the Cameroonian government is auctioning off to international companies certain areas in the rainforest where they log. This is an Italian company, they're logging for timber there. So you have networks, of course, where they have to bring out this timber to the next port. And the next port is more than 1,000 miles away to the west. So you have all the roads where there's a constant stream of trucks bringing out this hardwood and timber to the coast. So you have networks again. Secondly, you have a lot of people coming into that area to work for the logging companies, first of all, but also to provide services for all these truckers. Mind you, there are no supermarkets or anything there. It's just bush tracks, and then you have little settlements like these. And the people live exclusively of bushmeat. These are pygmy hunters. This is a Bantu woman slaughtering a duiker. And we were there for almost 3 weeks, and you couldn't get any protein except from bushmeat. So we were eating monkeys, we were eating duikers, we were eating snakes, we were eating porcupines. So the whole range of animals you had in the forest, you were eating. And all of these people were eating them there. All of these people were, in one way or the other, coming in close contact with wildlife, either through slaughtering it Here's a preparation of a monkey over fire. So you had constant exposure to wildlife and constant opportunities for viruses and pathogens infecting these wildlife to jump over into humans. Now, there are doctors. We went there with a Cameroonian army medic, and he's working with a team of scientists in southern France, but also here in Philadelphia with Beatrice Hahn. And they are actually monitoring these populations. First of all, what you see is you have very high HIV rates. Because the truckers, the HIV spreads along the trucking routes. The truckers, of course, have lots of relationships with the population there. So that's one way of HIV to spread. It's up to 25%, so almost every fourth person has HIV there. But they're also analyzing blood samples, and what they're seeing is they have indirect evidence of transfer of pathogens that normally only infect monkeys and apes, other than HIV, related to HIV, but other than HIV. They have indirect evidence that these populations have these viruses through antibodies, and also some of the people actually are infected with novel pathogens. They don't make them sick. They don't pass them on yet. But we don't know whether that happens, if that happens, but the first steps are already taken. In a nutshell, this is one of the areas which Peter pointed out in Central Africa, where you have all the drivers there. You have mobility of population, you have a population growth through an input of population working there. You have a very close and intimate contact with wildlife, and also you have networks that can bring these pathogens to the big cities in West and Central Africa. Now, I'm a biologist, and what I realized there is with chimpanzees, it's almost, if you look at evolution and the factors that drive evolution, and also the selective factors that are going on. With HIV, it was like HIV found itself in a chimpanzee population that was dwindling. And any virus that is at the moment in chimpanzees has a population of, well, there are maybe 250,000 chimpanzees left. Of course, for any virus that makes or manages to jump into humans, it has an ever-expanding population of 7 billion people, so the selective pressures are there for viruses to adapt to its new host and to go into a human host and to survive in a human host. So listening to Peter, I find it very interesting. Some of the efforts are going on there to understand which viruses are circulating, which viruses can make the jump into humans. And of course, if we can prepare ourselves, if we have genome or genetic sequences of the viruses, that would be a huge advantage to have vaccines in the future. And these areas, as remote as they may be, have to be on our map so that we can react early enough, or we can actually act preventively before it's too late. Finally, I just want to say with Ebola, it was basically the same thing. Ebola, the present outbreak in West Africa, originated in Guinea, and it was, again, they call it the forest region of Guinea, but this is not a forest region anymore, it's a deforested region. There has been a lot of deforestation going on. Again, from an evolutionary perspective, if this virus finds itself in bats, and the habitat of bats is being destroyed, then for the bats there are two ways to go. Either the bat population dies out, or this bat population manages to live next to a human population. Bats, of course, are mammals. They are very adaptive in that sense. And that's probably what happened with Ebola. You had, again, the bats living in very close proximity with humans, and that made it very easy for this Ebola virus to jump from bats into humans. And then, of course, it was in an area where the next capital cities were never very far. So in a way, with Ebola, everything was at place, the same as with HIV, everything was at place. We know these factors, and it's probably a matter of looking very closely and thinking very hard how we interact with nature, and thinking also how we can use our knowledge to prevent it in future. Thank you.
Thank you, Carl. Amy Maxman is going to share some of her reporting on Ebola. Amy's also a science journalist and has been supported by the Pulitzer Center.
Hi, so I am a science journalist. Thanks a lot. And I'm going to talk. My talk's a little bit different. It's going to be a lot less sciencey, actually. So I'm talking about our being the U.S. response to epidemics that happen in the developing world, and also what we respond to and what we don't respond to. So I had gone to Sierra Leone to cover the Ebola outbreak, and I was going to just cover science because that's what I do. I have a background in science. I wanted to cover virology and treatments to it, and anthropology. But what I was really stunned by there was the large distance between the response as we see it, if I just do a Google image search, versus what you see on the ground. And I was very intrigued by that. And so that's what I really spent a couple of months there reporting on. So here, this is what maybe we think about: white tents, biohazard suits. Where I spent a lot of time was the hospitals, which is where people go when they're sick if they go seek medical care. And that's before the international response shows up. So this is just a photo of exhausted nurses. So if you go to public hospitals, what you'll find are underpaid, overworked staff. Often there's no electricity, there's no running water. So specifically, I spent a lot of time in the west of Sierra Leone. There's a large western district on the border of Guinea. And in April 2014, that's when Doctors Without Borders was shouting a lot about how there's a really terrible Ebola epidemic or an outbreak that at that time it wasn't huge, but they were saying this is a bit different than what we've seen before. They were worried. And at that time, Dr. Khan, who you might have heard of, he's the leading virologist who was in West Africa. He was at this big hospital in Western Sierra Leone. Is it not loud enough? Sorry. So he was at this hospital in Western Sierra Leone. He's worried because they're on the border with Guinea. He calls all of the young doctors he's trained in the country to come there. He convinced a lot of nurses to come for a training on what should they do should Ebola showed up in Sierra Leone. They came, but within a few weeks, there was a woman who was pregnant who came to the hospital. She miscarried, and she couldn't stop bleeding. She kept bleeding. They moved her to the surgical ward. He knew enough to test her not only for Lassa fever, which is a hemorrhagic fever that's endemic in that area, but also for Ebola. She died, and then she tested Ebola positive. At this point, it blows up in this region. Ebola is just spreading very rapidly, and the nurses have to care for it. They don't have gloves. They don't have protective gear. Commercial shippers stop flying, so it's hard to even get protective gear into the country. So they're using tarps to cover themselves. This is just one of the walls of the hospital when I was there. It's just plastered with flyers for memorial services for nurses and ambulance drivers and lab technicians who've died because they don't have protective gear and they're working very hard to do what they can. So May, June, July, it keeps escalating. This is one of the morticians from the hospital, and he convinced me to come into the morgue. Ebola was not so strong in the area when I was there at this point. But during the peak of the outbreak in this region between July and September 2014, there was more than 3,000 Ebola tests done at this hospital, and he just slept. He showed me where he slept right on the other side here of this bed. He slept on the ground because around the clock he needed to prepare bodies, and there was just piles of corpses. By the time I got there, so if you think August, in August they're dealing, and this is in the West, they're dealing with expanding. There's 1,000 people that had died by August 2014. That's when the World Health Organization decided there was an international health crisis. That's when fundraising begins, and countries start donating lots of money. But that even takes a lot of time because countries decide how much they're donating. They decide how are they going to implement it. Are they going to get the U.S. Army to do something? What will they do? This all takes time. The whole time, the virus is just multiplying and spreading, and it hits the capitals. By October, there's 3,000 people dead. And still, there's not a lot of international either. So all of the white tents and the biohazard suits you're seeing, those are starting to show up around then. Late November, early December, that's when finally, I think mid-December, they decide there's enough ambulances and these white tents, Ebola treatment centers for people. By early December, there's 6,000 people dead. I come then. When I come, there's like the plane is full of aid workers. And at that point, when you go to public hospitals, what you'll see is there were strikes across the country, both in Sierra Leone and Liberia, I think also in Guinea. And this is just some of the graffiti. It says, "Pay my day, please pay us all of ours." So part of the problem was when all of these staff were working, they were told they were going to be paid $50 to $90 a week for risking their lives and going through this tremendous thing. And either some people were paid, but a lot of people were not paid, or they were paid half, or they were paid extremely late. So there was protest just before I arrived. Somebody had dumped 3 corpses on the ground just in protests. And part of the anger was also, headlines were reading, Obama donates $100 million. So there's huge donations. It's clear that people are donating a lot of money. There should be a lot of money. Why aren't they getting it? And you can see that there's money too, and this is in December when I'm there. There's United Nations helicopters flying back and forth. There's parking lots in the capital cities that are full of new cars that are donated. They've got plastic wrap still on them. And there's things happening. There's Ebola treatment centers are being put up. And there's lots of donations. And it's clear some of those things are going to very good purposes. Other things are a little bit questionable. But what people keep asking me as I visit various public hospitals is who's eating all of the money, because they still were not getting paid. So that sort of became my question too. This is just a chart from May 2015. So these are donations, so not just even promises. These are actual donations made to West Africa. The US was the biggest donor with 1.5 billion. You can see the UK has 330 million and so on. But here's the thing for how donations are made. You might all know, I didn't know this until I looked into it because I have a science background. So when bilateral donors like the US, the UK, when these governments make donations, they make donations and their money goes to either the United Nations agencies, to their own agencies like the CDC, or to big NGOs like the International Red Cross, like Save the Children, like Doctors Without Borders, the big NGOs. And then there they might hire contractors who then hire the next level of people, that maybe they hire other NGOs. There's a whole hierarchy. Where it does not go is like to the public hospitals or to nurses who are within the system. It doesn't go to the government of the country. It goes outside of the system. However, the government of Sierra Leone, it's an extremely poor, one of the poorest countries. They asked for money to help them cover hazard pay for health workers in their system, and the World Bank stepped up and they did donate money towards it. And so in an investigation I did, less than 2% of the 3.3 billion was earmarked for all of those thousands of staff who are in the national health system. So a very relatively small amount. So I won't go into all of it here. If you'd like, you can read my piece, but I kind of became obsessed with those. So at least 2%, why are people still not getting paid? What the World Bank did is they asked one of the UN agencies, the UN Development Program, UNDP, to implement this kind of awesome way of paying people through mobile phones, because it's a cash economy. Very few people have bank accounts. You get a text message that says you have $90, and you go to a vendor and you pick up $90 in cash. However, super smart idea. It's not simple because you still need a system. You need like administrators and human resources departments who keep track of who works where and when they work there and things like that. So I went about trying to make an Excel file, at least of like at this one hospital in the West, who works there, who is owed which money. In the end, I guess a sort of sad conclusion is I don't think anyone really cared if the system worked or not. Because I don't know if there's a lot of if the system doesn't work, I don't know if it ever comes back to the people who are told to implement the system. So that's sort of the end of that. But why should we care? That was Ebola. Ebola is over. And I think the reason why it bothers me now is because it's just sort of a question put out there. So that was Ebola. We hear there's definitely going to be other epidemics that are happening. What would have happened had in April, when Dr. Khan knew that Ebola might cross over the border, what if there was a hospital with running water and electricity and gloves and protective gear and nurses who were supported and also paid a living wage? What would have happened then if they were prepared to take care of the outbreak themselves? As you know, when we had a case in Dallas, I think the CDC might have dropped the ball for a minute, but nonetheless, there wasn't much of a spread of Ebola in the U.S. So this is a photo of, they did, so this was this woman with the hair cap. She was the head of the Ebola treatment area at the hospital, and so they built a new nurses' quarters for her. So I visited her in July, and this is the nurses' quarters for the isolation unit that's there for Lassa fever and other hemorrhagic fevers. It's the rainy season. There's like a soggy mattress in the corner where nurses on the night shift can share that mattress. All 15 of them have to huddle in there when it rains. Their files are completely covered in water and runny. That's the state of the public health system that's there. And so it may be a challenge and a question for our foray before and after these crises is there a way to support the actual systems in the places as opposed to just systems outside of it? So I'll end with that. I have an ebook that tells more of the story I talked about that you can download, or there's a sort of shorter version on my website that's for Newsweek and other stuff too. Thanks so much.
Thank you to all our speakers. Now that we do have a little bit of time for discussion, so what I'm going to ask people to do, first of all, if Sonia has questions for the panel, obviously she gets to start. But if you're interested in asking a question, the mic is just here. Please, everyone, because we have limited time, just if you can make your questions as brief as possible and indicate who on the panel they're indicated to, if that's appropriate. Thank you.
I don't think it's on.
Is it working? Okay. Concerning trying to identify these 300 million mammalian viruses, as we know, there are many viruses which are non-pathogenic. As a way to sort of parse that down to make it a much more economically productive thing to do. Also, we know that there are non-mammalian viruses that can also cause disease, and does it make sense to really just limit ourselves to mammalian viruses? Second question, comment sort of question to Ms. Schock, concerning the water thing and cholera in Lower Manhattan in the 19th century. You know, that was started, it wasn't that they were interested in supplying water, they were interested in developing a bank. And they applied to the state to get a charter for a bank, but the deal was, and Aaron Burr was one of the people who was involved in the development of the Manhattan Water Company. Aaron Burr was one of the people, and the deal was that in order to get a charter for the bank, they had to develop a water system, and that's how it happened. So they weren't really interested in water at all. My point is, I think it's the profit motive and greed that drives a lot of people here, and I don't see a solution to that unless we change our whole society in the way culturally we do it. I think we're so driven by the profit motive. You know, Winston Churchill said Americans can try everything first and then eventually they'll do the right thing. I think it applies to human beings in general. Human beings will try everything and then eventually they'll do the right thing. I don't know how long it's going to take for us to get there, but I'm somewhat cynical.
Thank you for that comment. Do I
You go first.
You go first. It's your book.
Yeah.
I mean, I think it is a huge political challenge to try to, and that Aaron Burr story, thank you for pointing that out, but I go into some detail about that in the book if anyone's interested. It's a great story. But certainly, right into, okay, sorry. The other one, that's the problem. Okay, there we go. Okay. Certainly the political challenges, I think, are huge. And, you know, we talked a lot in the panel about pathogens that are coming out of sort of distant parts of the world. But another point to keep in mind is it's happening here too. And a great example of a political challenge and emerging pathogens that are coming out of our inability to overcome these political obstacles are antibiotic-resistant bacteria, where we have known since 1940 or some odd that if we use antibiotics in ways that are not medically necessary, we will get these super resistant bugs. And yet, decades later, for decades, we have been using, the majority of the antibiotics that are used in this country, 80%, are used for commercial purposes. They're not for medical uses at all. And this is obviously contributing to our increasing problem with antibiotic-resistant bacteria, and yet we've only taken the tiniest baby steps to addressing that. So I think the political challenge on our side is, the rise of private interests. But the political challenge, like what Amy and Carl are talking about, is building these health systems in places that, where some of these pathogens are really most likely to come up. So I think it's going to vary in place to place, but I think the political challenge is obviously, I think it's one of the top ones.
Yeah, great question. I'll continue in reverse order, so I'll answer your second question first. Why mammals? Well, if you do the math on the previous emerging disease events, the vast majority of the zoonotic viruses, the viruses that come from animals, the ones that go pandemic, tend to come from mammals. So if you cover mammals, you cover the vast majority of future pandemics. So that's why we chose mammals. You should probably include birds too, because of avian flu being such a threat. But we've done the math on that. It doesn't increase the cost too much. The first question is more difficult. You're right. It's a stamp collecting exercise, but some of those stamps are penny blacks, and some of them you've got to throw them out. Some of these viruses will be killers. Some of them won't. How do we work that out from a viral sequence? It's not straightforward. So as an example, first of all, we're only looking at viral families that include those that have gone into people from animals. So we narrow it down straight away. Then when you get a sequence of a virus and it looks like a relative of a known nasty pathogen, just like we did with SARS, we found other coronaviruses in bats, a whole host of them. Some of them looked very similar to SARS. So we sequenced the spike protein, the protein that attaches to cells. Then I didn't do this work, but my colleagues in China did the work. You create pseudoparticles, you insert the spike proteins from those viruses, see if they bind to human cells. And each step of this, you move closer and closer to this virus could really become pathogenic in people. So you narrow down the field, you reduce the cost, and you end up with a small number of viruses that really do look like killers. Then you look in people and you say, in the people that live in the region where this animal lives, that are exposed to that virus, do we see antibodies specific to that virus? There's the first evidence, just as the groups out in Cameroon are showing, of a new virus spilling over into people. What are they doing? What's the high-risk activity? Can we find an alternative? So it's not straightforward, but there are ways we can hone it down. And I think we've just got to do it. It just makes sense economically and for humanity's sake.
I have just one comment on the absurdity of things. I asked the company who did the logging in Cameroon what the wood is, the timber is used for, and it's mainly used for matches and toilet paper.
Oh,
so, on that note.
We're not going to have a lot of time to go into this, but if you'd like the details on precisely how we do this in a laboratory, I'll be happy to talk with you about it afterward.
Thank you very much for talking about the most likely origin of the next pandemic, but perhaps the highest risk. Any comment on biohacking or intentional man-made pandemics?
This came up really after 9/11 when people became quite concerned when we had the anthrax attacks, and there was a big emphasis in developing a program nationally here called which was focused really on biodefense. It was subsequently expanded to include emerging infections because it became clear that the majority of emerging infections were not by our design, but in fact came from nature. There has been a shift back now again toward consideration of deliberate biological weaponeering. I still don't feel, but that's all I can say, it's a feeling on my part, that this is a huge risk, because it's easier, frankly, to just find these things in nature. But it is something that we're all concerned about, which actually then leads me back to addressing your question anyway. So one of the things that what we try to do is first find out whether or not a virus can bind to a cell, because viruses only grow inside of cells. So that means it has to attach to something on the outside of a cell called a receptor, to which it binds, locks, and then enters. The next thing that the virus has to do when it enters a cell is to find some way to evade what's known as the innate immune system, which are molecules that are released inside of cells that basically decimate viruses so that they can't otherwise grow. So we look for these evasion molecules, these evasion systems that some of these viruses have. So if you have a virus which has the ability to get into a cell and to evade the innate immune system, then you know it's a serious candidate for causing human disease. And typically, the closer the host organism for that virus to a human, the more likely it will be to be able to infect a human and cause disease, which is why we focus so much on apes and a lesser extent on other non-human primates. But that's not necessarily the only indication of disease. So for example, with West Nile virus, its ability to replicate inside of native corvid species here, in North America, allowed it to explosively grow and then become the source of the virus that subsequently gave rise to the West Nile virus epidemic. Now, the last speaker was talking about, about the Ebola outbreak and how, there were lessons learned from the Ebola outbreak in terms of rapid response and so forth. In 2004, 2005, Peter and I had an award together from Google.org that was designed to address proactively some of these things. And we actually set up outposts for this sort of disease monitoring. One was in Bangladesh. That system has become quite robust. The other one was in Sierra Leone at Kenema General Hospital. How ironic is that? And we actually placed into a hospital there equipment that was modern, that was set to go. The first thing that we ran into as we were trying to bring in the machinery is that when we hit Freetown, people sabotaged the equipment because we didn't have bribes. And the United States government will not allow us to allocate money for bribes. So we then had to bring in the missing parts, in personal luggage thereafter. After 3 or 4 years, Google decided that they no longer wanted to support this particular organization. So as a result, that whole thing collapsed. Now we have a recent request that came from the Obama administration to the Congress for $1.8 billion to address Zika. And the information that came down yesterday, or maybe it was today, we're not going to give you any money for Zika, spend the money that we gave you for Ebola. So the major problem that I see in much of the world is poverty and lack of infrastructure. And until you can address poverty and lack of infrastructure, even though we've got great materials and great technology and so forth, it's very, very difficult to deploy. I mean, you see more of this than I have.
Yeah.
Poverty is poverty, and as you said earlier, it's lack of clean water, cholera and everything else. So poverty is the big enemy, I think.
I just had a quick question about prioritizing. We look at something like Zika, which was kind of at the bottom of the pile of things that we were worried about, and then suddenly we've bumped it up to the top. As we're discovering new viruses, how are we going to prioritize which ones we should be concerned about, which ones we should be trying to address first. Obviously, there's limited resources. We can't take care of everything, unfortunately, at the same time. So.
Well, let me say something about that. So first of all, if you look at the hotspot map that we produced back in 2008, prior to West African Ebola outbreak, West Africa's a hotspot. Prior to H1N1 so-called swine flu, Mexico is a hotspot. It's clear from looking at it logically, you can prioritise where to work. It's what you do there that matters. And Ian makes the point about our responses are always after the fact. It's just not good public health. We do good public health on smoking, diabetes. We do lousy public health on pandemics. We need prevention, not cure. So, great example. We've had the West African Ebola outbreak. $5.14 billion was used to back-fund a lot of the operations that happened during the outbreak. Where's the money now to move forwards? There's a global health security agenda. We need the funds to be allocated to the places that are going to be the next outbreak, not the one that was the last outbreak. And we know where they are, we know how many they are, and we know how to go about identifying them. I think it's good common sense public health to get out there and do this proactively. For prioritization, you go back to which wildlife species are these viruses found in? Are they in areas where there are already ongoing outbreaks of undiagnosed illnesses that could be those viruses? Are the people who are hunting those animals, are the people who are highly exposed? There's a whole series of things you can look at to prioritize, which are fairly logical. I just think this is not rocket science. It's straightforward. We just don't yet have the global will to do it.
Thank you.
So I'm curious, if we do manage to find these viruses and find places where they are jumping over to the human population, and now we know that, how do we contain that? I mean, knowing it is nice, but doing something about it seems nicer.
Well, let me give you one example. I'm sure other people have other examples. A great example of this, going back to the work we've done in Bangladesh with the ICDDRB, the old cholera hospital, actually. It was formerly cholera hospital with Steve Luby and his team. So we know that there's a virus out there called Nipah virus, which is really lethal. If it causes small localized outbreaks repeatedly every year. The average mortality rate, if you like, from people who get infected is around 70%. There are no vaccines, no drugs. It's a nasty virus. It's a bat-borne virus. These bats are fruit bats. And people in Bangladesh drink this sap that they scrape out of a date palm every evening. They put a pot up there, and the sap drips into the pot. They go up in the morning, bring it down, sell it off fresh. You can't leave it because if it ferments, it's forbidden. So it's got to be fresh. Unfortunately, bats feed on these open wounds on these date palm trees, and we now know that they excrete virus into those pots by urinating into them, spitting into them, and defecating into them. And occasionally you find a dead bat in a pot. It's not a good thing. But there is a simple solution, and the simple solution that ICDDRB are trying now is really cheap to make bamboo skirts that you can make very, very low cost. Put them around the place where you tap the tree to prevent bats getting access. It's all about reducing contact, still allowing people to do the things that they've done for thousands of years and make money out of them, but finding a solution that keeps the contact between wildlife and people low. And I think every virus is going to have a different solution, but we need to be creative and we need to find these alternatives. And ultimately, we need an alternative to logging the tropical forest in an emergency hotspot for matchsticks. That's not a good thing to be doing right now in the Anthropocene, in the age of pandemics.
And that's what Ian said as well, poverty. And I witnessed that during the Ebola outbreak and also in Cameroon from what I showed you there. It's sometimes a totally different logic at work. I mean, the beginning is the demand which is created through companies saying we want to do logging there. Next to poverty, corruption is the big thing, because you can guess that this logging company is having deals with the Cameroonian government. They say, okay, we designate this area, you can log there. And there the whole thing starts, because the people do not understand the core causes and consequences. They look for work, they look for opportunities. So you have all these people moving in there, nobody controls them, nothing. The pictures you saw during the blood testing, the blood is also tested for HIV, and I saw several people being tested positive there for HIV, and the people there die of HIV. They do not get any medication. The medication is stuck in Yaoundé or in Douala. It never makes it down there because there are corrupt forces at work which hold back the medication and then auction them off again within Cameroon. So it's a whole set of obstacles you have on the ground which you need to understand the logic in order to implement programs that can stop and reverse these things. It's very, very difficult.
Hi, I'm a neurologist. I came in late, but did you discuss why, with the Zika virus, the cases where Zika was isolated in microcephaly is in the single or double digits compared to the 4,000 plus microcephalics that were identified in Brazil? I know that maybe of 1,500 that they've identified, for every, let's say, one that they've identified as truly microcephalic, there are 2 that they've thrown out, but still the numbers where they've actually isolated the Zika virus is very low.
Well, the question is really what is the mechanism by which Zika virus infection might cause microcephaly. So there, first of all, by isolation, people typically mean taking infectious virus and pulling it out and demonstrating that they can grow it in culture. And the other way in which people try to identify a virus as being present in a sample is by demonstrating genetic material or proteins made by that virus and so forth. There hasn't been a lot of analysis of microcephalic brains because most of these children are still alive. And it may well be that if these children were to die, that you would find fossil fragments or genetic elements of these viruses present within those brains. I just don't think we have that information yet. Now, what people are trying to do is to develop indirect mechanisms of demonstrating Zika virus infection. At present, there are some methods that are called serology, where you look for antibodies, but they're not as specific as we'd like them to be, so you have cross-reactivity between that and dengue virus. The question I want to get back to, which I don't think we addressed properly, is how we're going to identify which of these viruses of the 320,000 that Peter and I described as being potentially there should be prioritized in terms of importance. We do a lot of work using high-throughput sequencing instruments to characterize unknown viruses that are present in the blood supply. And we've come up with very rapid and less expensive ways to do this. I have to say that in the analyses we've done thus far of blood, we haven't found thousands of new viruses that would need to be characterized. If we were to look at blood from areas like Cameroon, where we don't currently have access, and if anyone would like to provide us with access, I promise you we will analyze them very quickly and let you all know how many viruses are lurking there. But the number of viruses that would need to be characterized, and to that extent, I think would be number in the hundreds. And that's a fairly simple set of experiments to do. We can characterize them in culture. We can look for their ability to get inside of human cells. We can look for their ability to suppress the innate immune responses that would normally kick viruses out of cells. We can adapt them to organ cultures, and ultimately we can move them into animal model experiments. And if we take it through that whole series of entry points in this rational fashion, I would predict that we could identify those viruses which pose the greatest risk. And we could give you a top 10 list of what we would be concerned about in the Americas and in Asia and in Africa and so on, it would not be a staggeringly expensive process to pursue. But someone has to pay for it. You know, there is no way to do the work otherwise.
Okay, on that note, thank you all so much for coming. And the book is on sale in the back. We're also having a reception. Please do stick around and have more conversation. Thank you.
Thank you for joining. Oh, don't forget to buy Sonia's book on the way at the back. Thank you.
Well, you're absolutely right. No, you're absolutely right. Layer after layer.